Welcome to the Australian Biocommons webinar series. We aim to share useful information about the latest digital techniques, data and tools in these sessions. Each month we hear from our local and international peers who present a bioinformatics topic that we hope will support Australians to deliver their best environmental, agricultural and medical research. My name's Christina Hall and I'm the Australian Biocommons Training and Communications Manager. We particularly appreciate those of you joining us live today, and you'll have the opportunity to ask our speaker questions via the Q&A function on your dashboard. These will be addressed at the end of the presentation. This session will also be recorded, and you'll soon find it on our YouTube channel, along with previous recordings of our workshops and webinars. We hope you'll keep in touch to hear about future webinars via the channels listed on the screen here. Now, before we start today, we'd like to acknowledge the traditional owners of the lands on which we work and pay our respects to the elders past, present and emerging. With us today is Associate Professor Parwinder Kaur, who is the Director of DNAZOO Australia and who works within the Faculty of Science at the University of Western Australia. Parwinder leads an innovative translational genomics research program that aims to translate fundamental science into ready-to-use solutions across the agricultural and medical sectors. Her DNA Lab team enables research to span the spectrum of scientific advances beyond the traditional lab-to-landscape model, using technologies such as CRISPR, single-cell and 3D genomics. With DNAZOO Australia, she's on a mission to provide genomic empowerment to unique Australian biodiversity, facilitating conservation efforts for threatened and endangered species. An innovator across a diverse range of fields, Parwinder has been awarded the prestigious Science and Innovation Award for Young People in Agriculture, Fisheries and Forestry by the Australian Academy of Sciences in 2013. And more recently, awarded the Microsoft AI for Earth Award in 2019-20 for DNAZOO developments. And this is the project that we'll hear about today. We're thrilled that Parwinder joins us to share how DNAZOO is facilitating conservation efforts through rapid and open release of high quality genomes. So welcome, Parwinder.
Hello, everybody, and thank you so much, Christina and the whole Biocommons team for inviting this seminar today and having me here. Okay, so I'll get started. So as Christina mentioned that we're going to be talking about DNAZOO project today and how, so I will introduce you guys to what is DNAZOO, what are the different workflows and technological advancements we are using at DNAZOO to be able to facilitate the conservation efforts through rapid and open release of high quality genomes. Before I get started, I would like to acknowledge this is a partnership between my lab, DNA Lab at the Faculty of Science at UWA, and Baylor College of Medicine, the Center for Genome Architecture Lab, which is run by Professor Aidan Lieberman, who's also one of the founders of DNAZOO. So let's get started. I hope I can move my slides. Okay, yeah. So before we start this presentation, on behalf of University of Western Australia, I would like to acknowledge that this campus where we are situated is situated on Noongar land, and that Noongar people remain the spiritual and cultural custodians of their land, and they continue to practice their values, languages, beliefs, and knowledge. And we are very grateful for this opportunity to be on this land today. All right. So the big question, I'm sure you guys have are aware of this or in touch with this, and that is probably one of the reasons that you are joining us today, because you do care and you are aware of the biggest pressing global challenge we are facing at this moment, that as per the UN reports early this year, we've got about 1 million species at the risk of extinction. And not just the risk of extinction, but also we are losing one species every 6 hours, which is never going to come back again on this planet. So it's a pity that we are here on this situation. We are here in this situation. Being humans, we've done some wonderful advancements in every sphere of life, but we've also occupied 96% of this planet, which also belong to many other billions of other species which live on this planet and co-inhabit that with us. So we do have a bit of a responsibility to that, and how we can support and help, especially because we are situated in one of the 17 countries which are described as mega diverse countries. Although there is only 10% of the global surface which is these 17 countries, but we support more than 70% of the biological diversity on the earth. So that brings a lot more responsibility to Australia to protect and support this amazing biodiversity, this rich and rare species, the part of the planet which we've got, and we're blessed to live on this. So what is Australia's situation in that is a little bit sad, because although we are famous for our terrestrial mammals, unique terrestrial mammals and the fauna, but we've lost more than 30 species, and this is continuing with more species going. I'm sure you guys know thylacine, big-footed bandicoot, and many other species have become already extinct, and many others, amazing ones, which we love, absolutely adore, are going on that path. So that is not a great thing for a nation which hosts the best unique biodiversity, especially our mammals. 87% of our mammals are unique or endemic to Australia. And that was one of the reasons for DNAZOO to sort of venture out and start this whole initiative here. And we know that many other are threatened and, as I said, are in danger of extinction. Australia leads the world in mammal extinctions. I hope we can change that very soon, as I see many different projects come on board this year, last year, like OzMammal Genomes, OzArg, GAP, Genomes of Australian Plants. They're doing some amazing, wonderful work and nationally connecting these resources. So I think we've got a great future. But at this moment, this is a situation we're sitting in. And then nature isn't helping either, because it's come to a point that we probably disrupted the ecosystems of the food chain so badly in this whole climate change that has been very, very obvious and prominent especially for Australia early this year, when we had a record-breaking 8.4 million hectares of land burned across Australia this bushfire season, an area which is larger than Scotland. It was massive. It has caused many species a big stress, but then also we've lost a staggering 1 million animals, and probably still counting how many more are gone and left us. Not just animals, but also the vegetation, the food they eat. So it's kind of the whole ecosystem has gone disrupted. And it's not just Australia, it's the wildfires been going around the world. And I got this figure, and I was shocked to see that there's a total area of 400,000 or more, which has been under fires in 2018 as per the data collected by NASA. So some big red flags out there. Yes, there are a lot of reasons and problems: habitat destruction, overhunting, toxicity, climate pollution, climate change, lots of problems. But today, we're not going to talk about problems. We're going to talk about what we can do in our little domain to be able to get started, take the first step, and then I'm sure the path will start to appear. So being in a genomic space, what I have realized, and I worked with many non-model plant species and other species across the tree of life, the critical barrier to research has been the lack of adequate genomic resources, and always trying to work with these model species, which sometimes even don't have that problem. So we're kind of manifesting everything into that and relying on that model species to show us a path, which is not a very fast solution, because it takes long time to translate that research from one species and take it to another, and then look into that. And I'm sure I don't need to go into the detail of that. And let's see that how and this is not just us, it is the entire world faces that problem, but these are some of the wonderful people that I have met who have shared an amazing expertise in the 3D genomics, transcriptomics, genome assembly space, but also share this common pain of losing our species. And that became the launch of DNAZOO, which happened in November 2018 in Houston. Professor Eris Aidan Lieberman here, who's also the inventor of high-C genomics, and is one of the founders, and with this lady hiding at the back in the blue shawl, Dr. Olga Datchenko, who's written the 3D DNA workflow, which we will go through today, and which has become one of the most critical things to be able to automate genome assemblies and fast forward them at a very cost efficient way. So these are the co-founders of DNAZOO, and this is our global consortium, which we started with, and of course it has grown, and there was many people who were missing in this meeting as well. But it has been a real amazing collaboration network who all share the same passion. And when we talk about passion, I mean, I have been always intrigued by the evolution of life and how it started billions of years ago, and where all this enormous biodiversity originated, and where is the basis of it, or how can we track it back? Can we actually go back in the time and see what would have happened, how this would have come to a state where it is today? So that has been one of the key reasons that I got connected to these people at DNAZOO Consortium, because being motivated, we shared this motivation to solve the two important mysteries in biology. And most of us have used tracking genomes in space, time, and in single cells to be able to resolve those mysteries. So today we will only be talking about tracking genomes in space. And what I mean by that is, I'm sure that most of you get intrigued by this fact that how genomes get packed in that very, very small space. We call it nuclei, right? So human genome, let's talk about human genome. Approximately 3 billion letters it is written in. And if you stretch it out from one end to the another inside one nuclei, it's about 2 meters long noodle. And this 2 meter long noodle sits in a 6 micron nucleus, which we can't even see with our eyes. You have to have a microscope to be able to see that. So how does that really happen, that all this is packed in that tiny space, and then have all that structures, which we call chromosomes, and how does that happen? And that has been studied extensively as part of the genome architecture. And there are lots of technologies which we can use to do that. But that was one of the key reasons that gave birth to DNAZOO technologies, which we use today. So going back a little bit to the challenge, the long-standing challenge, that why we are not able to study non-model species as we study the model species, and what are the genomic resources we require. So normally we would like to read our genomes as this nice karyotype of chromosomes, but we end up with reading lots and lots of short bits and pieces of that 2 meter long noodle. Yes, we've come a long way in technological advancements, but still we do not have any technology that can read all that 3 billion letters in one go without any problems. So maximum we can read there is a limitation to that. So how can we actually convert that from this state to this state, so that we can ask some meaningful questions in a non-model species, which has probably figured out how to live in a desert environment, like our cactus mouse, or which has figured out how not to age and always stay young, like marsupial mole rats. So how can we do that? So that so I'm gonna take you back to the human DNA, where yes, we can assemble those 3 billion letters, but it costed us $2.7 billion and many years to get that right. Yes, we've come a long way, and now we are able to do a $1,000 sequencing cost for a non-model species, completely de novo assembly at DNAZOO. And that is the journey which we will be taking a closer look today. So what happens? You got this 2-meter long DNA, chop it into many pieces, make a library, and then you read it on a sequencer. You get lots and lots of reads, and bioinformatically, all we trying to do is we tile those reads and then get a consensus contig, right? I'm sure you guys are all with me. We're diving into now the whole complexity. But this works perfectly fine. No problems with that. We got computer sources. We can do all that, billions of reads these days, tiling process. But what happens when we hit a repeat? That's where it gets disrupted. And that is one of the key challenges that assembling genomes has been a very, very difficult task. So that we describe in the world, in the genomics world or the genome assembly world, we call that the scaffolding problem. And how that scaffolding problem get resolved, there are a few ways to do that. What you can do is you can order and orient contigs using linking data. That linking data could be your long reads. Like, for example, you generate lots and lots of short read data with Illumina, which normally everybody is doing. And then you go to Nanopore or you go to PacBio to get longer reads so that you can orient and link those contigs. But still, to get to a chromosome from millions of contigs is not that trivial job. And it again ends up taking institutional consortiums, lots and lots of dollars. And or if you don't have that, then you end up having all these different sizes of contigs. So that's where Hi-C is a very, very powerful source of linking data. As I said, Professor Aaron Lieberman invented Hi-C in the first decade of 2009, the major paper that came out and was a revolution was published in Cell. But what is Hi-C and how does it work? So I've had the opportunity to work with his lab in July 2016. That was the first time I visited his lab, and I was chasing a dream of assembling my small little Australian key pasture species, clovers. If you know clovers, I'm not sure, but that was my postdoctoral fellowship, and I was chasing it for a few years. By the time I had already used all the major technologies, worked in all the leading labs, including BioNano, which was one of the key ways to do the chromosome length assemblies, working with Professor Jaroslav, used PacBio, used pretty much every technology, but I still had 84% of my genome assembled and had lots of gaps where most of the important genes which I was chasing were sitting. So he kindly let me in his lab, and we worked on clovers, and we tried to put that jigsaw puzzle together. And it was an amazing experience. And how we use Hi-C to put that jigsaw puzzle together, I'm going to take a quick Facebook example to show you guys how does that orthogonal approach or the contact mapping works using the Hi-C data which we generate. So how it happens is, if I give you a puzzle to solve, and I take your box away, which shows you how it should look like at the end, and also blindfold you. So you have absolutely no idea. You've got these, probably, let's say we are doing only 300 pieces puzzle. So you've got lots of pieces in there to put, and you can't really see it. You can just feel it. But how about if I take your blindfold away? Then you can visualize it, right? So visualization has a great power. And let's take an example and try and understand that. We're going to be stalking Homer Simpson Facebook page, and we're going to go through all his photos, okay? So what are you going to see on Homer Simpson's Facebook page? Lots of selfies. We take a lot of selfies these days. So you'll see a lot of Homer on his Facebook page, obviously. You'll see Homer with his wife, Marge, in some of the photos. Of course, with his kids, Bartley's and Maggie. You'll see his neighbors, Flanders, because he does come in contact with them quite often because they live in the neighborhood, and probably many other friends and family members which he hang out with. So this is how a visual map of somebody's Facebook page look. And you must have noticed that sometimes when you post a lot of photos on Facebook with somebody, it comes back with a suggestion to you that you might know this person, or you must be related to this person or something like that. So Facebook uses orthogonal mapping approach at the back end. And how about we make a Facebook of genomes? So what do you need to do for doing that orthogonal mapping? You can do that using the context frequency. So here I'm building a symmetrical square, and you've got on your x-axis and y-axis exactly the same characters, the characters who appeared in Homer's Facebook page in those pictures. Okay, and you've assigned a number in that symmetrical matrix to every time those two characters come in or appeared in the picture together. Okay, simple. Let's keep it very simple. But having said that, let me remove your blindfold to solve this jigsaw puzzle and overlay this with a heat map. So the moment you put a heat map on top of the numbers, we very quickly are able to see an enrichment square in here. And that enrichment square actually tells you about the family structure of Homer Simpson. So you can identify or map the family very, very quickly, because they were in contact with each other at a much higher frequency as compared to they were in contact with any other people or characters in those photos, right? You can even tell that Homer and Marge are related, or husband and wife, because they appeared in the photos at the highest frequency. And now let's go back and imagine our chromosomes. You're thinking, yeah, that was the idea. I get you started thinking. So let's go one step backwards again. So how can we take a photo of all the bits of the DNA which were in contact when they were packed inside a nucleus? Because if we can take a photo of those bits or the loci which were sitting together in the chromosome, can we assemble chromosome length genomes? Yes, we can. How? We can map those contacts, and the bits or the loci who are sitting on the same chromosome will obviously have much higher frequency, or the contact matrices, as compared to the bits or the loci which are sitting on different chromosomes. It's a very simple principle and a very clever way of assembling genome. So let's go to how Hi-C helps. So what Hi-C does is in situ Hi-C, which I'm talking about in here. You cross-link your loci while it is inside the nucleus. Okay, we use formaldehyde for that. And once you cross-link, you use a pair of biological scissors to cut those bits of the DNA. And then you fill in with a sticky biotin enzyme, you ligate them, very simple. You just, follow your nose, pull those contacts out, and you sequence that. You generate millions of reads with that. With those, so these are your contacts or the photographs of the genome of the characters which were sitting together on that Facebook page. I hope I made that clear. So let's make a symmetrical matrix of a human genome then. Your characters here are your chromosomes, right, on x-axis, as well as on your y-axis. And then you got this symmetrical matrix, exactly the same way we did the Homer Simpson symmetrical matrix, but this one has actually got your DNA photographs, or those loci which were in contact with each other, and it overlays those heat maps for you, which are really nice presentation of your chromosomes. And you can take it to any resolution depending on how many million reads you generate. And we normally, for our genome assemblies, we do about 250 kb resolution, because that's more than enough for you to assemble a chromosome length genome. So yeah, it wasn't long that many people realized that Hi-C could be used to reassemble known genomes, and that's where the starting point was. There was a lot of publications that followed that 2009 paper, but then still there was a bit of a question that can you actually also do unknown genomes using the same technology? So yeah, there's more papers that came out, amazing crop species and many other animals, difficult that you could use, I see. But most of the time, or even today, I see people follow the same strategy that Hi-C is one of the other technology that has been used. So people normally go spend big dollars chasing that perfect assembly and have like bit of everything that is available or they could afford with that project budget, and Hi-C is just one of the things that is used in those things. So it doesn't still resolve. Yes, the problem of assembling genomes resolved, but that was resolved at the human genome time as well, but we haven't really made the proper use of the Hi-C to reduce our cost until we got hit by the Zika epidemic. And this was the first time that a genome assembly problem made it to the front page of, probably the only time the genome assembly problem made it to the front page of New York Times. And there was no genome for the carrier of the Zika virus, which is the yellow mosquito, which we all know. And that is the time I would like to introduce you to Dr. Olga Dachenko, that she took the challenge to work on this one, to resolve that genome into, which was sitting in millions of pieces, into a chromosome length scaffold in a cost-effective and an efficient way. So what was required was something automated that can put those pieces together once you've done the Hi-C experiment. And I do not have a video to show you the Zika one, but I do have a video to show you that how we assembled Medicago truncatula genome, which has used already all the technologies, including PacBio, BioNano, millions of short read from Illumina, paired and as well as made pair assemblies, but it was sitting in this condition, whereas there has to be 8 chromosomes. That's what a Medicago truncatula genome looks like. And how that was my first experiment in Aiden's lab, and how we use the 3D DNA suite, which Olga developed to resolve the mosquito genome, to do this genome. So I'm going to play a really fun video. Get ready for some awesome music. And this will show you how we do the assemblies. This one was timed, done under 8 minutes for the entire genome. Fun, wasn't it? It's like playing a video game. That's what my 7-year-old thinks, that when I'm assembling genomes that I'm playing a video game. But actually, this is how juicebox assembly tools work. And this is how the 3D DNA suite, which Olga wrote a few years back to make this dream of automated assembly possible. So this example, this is a small genome. It's a diploid genome. And it wasn't in such a terrible state that we could do this manually. But sometimes you have a challenge. So this is the Medicago truncatula genome, as I said. But sometimes this is your Zika, so mosquito, sorry. So this is the state it was in. And this is the 3 neat chromosomes that was actually required as an end-to-end assembly. And that was the key motivation for the whole 3D DNA code to be written, and also use Hi-C as a standalone with short read sequencing only to do the most cost-effective genomes, which we call $1,000 sequencing cost genomes, as we release through DNAZOO every week. So this is the state. This is where you need to be. Probably you can't do that if you want to just do that manually, how we did Medicago truncatula. You need an automated workflow. And this 3D DNA workflow, just box assembly tools, has been completely automated, and the whole code has been shared on GitHub pages. So please feel free to go and have a look. And that challenge was solved. Of course, that the whole rendering of that experiment, like how you see that we are doing that happens in the juice box or the juicer tools, which were written by Dr. Neva Durand in 2016. And this is like, again, a whole automated process. And I got really excited that juicer, high C must be a really awesome drink. I must tell you, I did try it a couple of years back. It's just too sweet. I don't know how kids handle it in the US, like it's the amount of sugar that goes into that drink. But it's, otherwise I love high C apart from that drink. Okay, so now we've gone through the method and we've understood that how DNAZOO workflow really works. So basically it's a combination. We mostly use, for $1,000 genome, sequencing cost genomes, we use only short reads. And those short reads, you generate about 300 million reads using DNA sequencing, the normal PCR-free libraries from Illumina. And then you do the contigs from those, but then you overlay that or render that with the high C data, just 100 million reads. Sometimes we do generate 200 million reads depending on the size of the species. I'm talking in general these numbers based on a mammalian genome, which is about the size of the human genome, or a koala genome, or a quokka genome, you can call it, for a de novo assembly. That's kind of the pipeline we use to do that, and we use the 3D DNA suite to assemble it all together, as I showed you. So what are the applications, and what we have been up to using all these tools at DNAZOO? We have been doing end-to-end assembly of clades. So that has been one thing we have been a little bit, that is one of the ways also that we have been prioritizing samples. Of course, the first thing that when prioritizing a sample comes is the conservation status for the species, but it is always fascinating to understand the clades and how the evolution happened. As I said, most of the people behind this have been really, really motivated by understanding evolution. So we have done that. We have done end-to-end assembly of clades, and I'm here showing you some of the Diprotodontia species, our Australian species, which were assembled using the de novo $1,000 genomes pipeline. We also have upgraded a lot of existing fragmentary genomes. So when we started the DNAZOO in November 2018, there were only 23 mammal genomes which were on NCBI. So we have upgraded most of those mammal species we could get samples for. So these are all the upgrades. So we've always, like, if you have a fragmentary assembly, which is really, really crucial for your research, and you would like it to be upgraded using high C, please get in touch with us on that. We've also been producing end-to-end assemblies for every sample. So every genome that has been released by DNAZOO is a complete chromosome length assembly. So we don't want to release more fragmentary pieces out there, because normally when you want to do meaningful biological research, it gets really, really hard working with that. I know that some people say, if I can just get some SNPs, I can work with that. But when you align those SNPs to a reference genome, and if that reference genome is not a quality genome, the amount of compute resource you use, the amount of time you use, the people who are doing that work is not trivial. So it's really great to have a chromosome length assembly, and that has been our key mantra at DNAZOO as well. Having said that, we do use mostly the short read sequencing and de novo assembly using $1,000 sequencing cost, but it doesn't mean that we do not work with long read. Long reads are amazing, and sometimes they have some amazing data which could help you resolve those challenges further. So we have developed a new method where we can generate platinum level assemblies using long read PacBio data, and that has been demonstrated across the tree of life. And this is just one example which I am showing of the peanut genome that was done in 2019 last year. Using this data, you can actually do karyotype evolution. So I'm showing this an example here between a wombat and a Virginia opossum, where you can actually look at how the evolution, how the karyotype has changed from one species to another when they are separated by, millions of years apart. You can also do conservation analysis of 3D contact patterns using this. And this has, due to time constraints, I'm not going to go into the details, but please refer to the paper by Rao and Huntley, which was published in Cell in 2014. Of course, for doing this kind of analysis, you have to generate very, very deep, you have to go deep sequencing. And that is what was done for human versus the mice models which were used in this study, but it doesn't mean that you can't do it. You can do it for any species of interest that you would like to. But yes, it will not cost $1,000. It will cost you more because you have to go deep into sequencing those contact points. You can actually study evolution at a chromosome length, a chromosome scale, and that was very beautifully described in the Dripton paper, which I talked about, the Zika. So if you're intrigued by that, please feel free to read this paper, which was published in 2017, and that describes how you can use this data to study evolution at a chromosome scale. It's really wonderful to have chromosome scale assemblies. You can do so much with them. It's just phenomenal. So having said that, there are lots and lots of tools out there. I'm sure you guys have been using those, but we went through a lot of assemblers. Most of the softwares or the workflows which are used by the DNAZOO consortium are in-house developed, except for the contigging process. The contiggers, we've looked through many different contiggers, and we identified that the WT wrap contigger works the best for the genomes which we have been doing, and then you use 3D DNA and JUICebox assembly tools, and that gives you these assemblies which I have been talking about, and as compared to all the other tools that have been prevalent or mostly used. So we've compared that across, but this is the combination which gives us the best possible assemblies. The $1,000 assemblies are not, it's pretty much the same as the human genome which was released in 2001, which was $2.7 billion, and compared to $1,000 assembly. So you can see this is a comparison showing that on the human genome data. So DNAZOO, what we have so far achieved this week, National Science Week, we are celebrating a release of 150 chromosome length genomes using the workflows or the methods which I have showed you. These 150 genomes features 133 vertebrates, which includes 117 mammals, and many of our Australian mammals in there, 10 Diprotodontia species. The marsupials are also there. If you're working on those, please feel free, go check them out. There are 9 plant species, 6 insects, some mollusks, a couple of mollusks, and a flatworm. And that flatworm also comes from Australia. That's one of the beautiful collaboration with Dr. Neil Young at University of Melbourne. I'll talk about a little bit more about that. But yes, please go and check all these assemblies and amazing species on www.dnazoo.org if you have not done that so far. Moving forward, it's really important once you get a chromosome length assembly that you have defined the genes, and for defining the genes, it takes a lot more effort, and we are all aware of the computational power it takes, but also the other types of data it requires. So at DNAZOO consortium, some people are generating that, so that we're collaborating with the labs who are working particularly on those species, and they have provided the transcriptomics data as well. But to have an overarching workflow that works with all the assemblies that has been generated without having to deal with some species has that transcriptomic evidence, some species do not. We're working with Dr. Matt McCannis from University of New Hampshire, and he spent a lot of time and released genome annotations using the Maker. So Maker is an automated genome annotation workflow, which we all aware of. And please go and check out the latest release that happened on the 8th of July, where he has updated all the genomes that we have released so far, and now they have annotations available for those genomes using the maker control files, and the whole code has been made available on GitHub, so feel free to go and have a look. It's a very, very useful resource, and we've also benchmarked that how long or what sort of compute resources you're looking at per genome, which is very, very helpful when you are estimating your requirements for that project as well. So yeah, so that's great contribution, and Math Mechanics Lab has come on board to do that with DNAZOO. So we're very grateful for all these collaborators who've come together. So let's take a look at some of the Australian ones, which we have been recently doing here at UWA. So the Australian lemming, that was one fascinating species which we have worked with using the nanopore data, and with the museums Victoria, Kevin Roy. This was a collaboration, and I learned a lot about rodents from Kevin. So thank you, Kevin, for this wonderful collaboration, and also Oz Mammal Genomics, who've been leading this amazing initiative to bring all these species to empower these species, the Australian species. It was a wonderful collaboration, and we were able to release a chromosome length genome this year in June. Really, really important to resource for the ongoing conservation work which Museums Victoria is doing with many other collaborating partners. And they desperately needed a reference level genome so that they can do the population genomics, that they're resequencing quite a lot of these rodent species who've suffered this wildfires and lost their habitats. And it's been a bit of a challenge. So this genome did not use quite a lot of data. This was not a very typical DNAZOO assembly which we do, because the data was already generated on nanopore, and there was about 3.5 million nanopore reads which were generated, and we polished up the short insert size Illumina reads, created a contig, and then we generated some high-C data, and then those contigs were assembled using that high-C data. We used about 450 million reads to do that. This was more than the usual we normally generate, because we had less data from the nanopore, so we went a little bit higher for our high-C reads, and again using the 3D DNA. So this was the state of the contigs which we put together using the nanopore data, and then overlaying that with the high-C data, we were able to get very nice 24 chromosome length genome assembly for this guy. And hopefully now that will be used for the population genomics project, which Kevin and team are leading from Museums Victoria. The other one which we did with Kevin and team as well, the Australian desert mouse. This has been amazing, and I have been learning a lot about these rodent species, and especially comparing them with the other species that Math Mechanics Lab has been doing, the cactus mouse. That these guys don't drink water their entire lifetime, and they must be doing something really interesting, that how they're surviving without that, and there's some lessons to be learned there. So this is another interesting species. We're very grateful for collaboration with Oz Mammal Genomes, Bioplatforms Australia, and Museums Victoria for sharing this species genome with the raw data which they generated, and we were able to release a chromosome length genome later this July. This species sadly is already extinct in Victoria and critically endangered in New South Wales, and that was one of the reasons we really prioritized this genome and looked into all the data which was provided to us, 46x coverage on Oxford Nanomore and short insert Illumina data, which was provided, and that was overlaid with the high-C data, the 20x data, and we were able to get, again, 24 nice genomes from that draft assembly to this finished product, which we have already released end of July, as I said. this is one of my favorites, I would say. The Victoria's faunal emblem fairy Leadervedas possum, and very, very grateful to Professor Paul and Sasha and the entire team at Zoos Victoria for sharing this project with us and sharing the journey with us. And this genome has been released as well a couple of weeks back in early August. We've got so this for this one we had really amazing data resource which was already generated using Oxford Nanopore and Illumina NovaSeq reads, and then we generated the high C reads. We generated about 250 million, again a little bit over than what we normally do, like 100 million reads, but sometimes you require that for big genomes, and this one was a really interesting example from draft assembly to 11 chromosome length genomes in here. So yeah, so that was a really fascinating project for that species. But when we did that species, we thought, oh, we've released the Tamer wallaby genome earlier, the $1,000 assembly. So let's look at the evolution patterns, because these two guys separated about 55 million years ago. So this is a really nice little karyotype evolution, which when we compare the whole genomes of the two species, then you can see that how the chromosomes have broken over the time, and the evolution has derived these species. It's really, really interesting when you're able to actually go zoom in these epigenetic data points which are generated by high C, and how much you can actually do with this data following this. So yeah, thank you Monash University, Professor Paul and Sasha, for this collaboration. We really appreciate it. As I said, the only platform which we have done is it's a really interesting one. This is a parasitic platform which impacts over 15 million people in Asia, and there's no sort of vaccine or insurance from reinfection. It's a really deadly flatworm which causes such a big trouble in a big population in China, Vietnam, Korea, and the Russian Far East. And there was no chromosome length assembly. Dr. Neil Young from Melbourne University collaborated with us, and we have been able to publish a chromosome length genome for this as well. So please check out. This was the draft genome which was provided to us by Neil and team, and this and we have a beautiful chromosome length assembly come out of that collaboration as well. So please feel free to have a look. Yeah, we live in WA, so how can I not talk about Quokka? Yes, the Quokka project's been underway. We have decoded the Quokka, but we're waiting for Quokka's second birthday, which is going to happen on 15th of September next week, to be able to release this genome. And it's not just the Rottnest Island Quokka. We have also decoded the mainland Quokka to be able to see that how these almost two different looking species. Is there a speciation even that has happened in just 22 kilometers distance? Or there's so many interesting things to look at with these cute little Quokka species with the star powers. But having said that, we've also gone a little bit further in love with Quokka, and there was this crazy idea of going from DNAZOO to DNA pool. which I explored with Microsoft, and they were really keen on supporting this and taking it forward. This could be a way forward when there is so much data being generated, and there's cloud computing and cloud resources available, with all these other technologies that come on board, that we might be able to use this concept of using the scats of different animals to actually do species monitoring, which is non-invasive, and which can not just track the health of the animal, but could also track the habitat of those animals and how they're doing, like health-wise and things like that. So there could be so many advantages, but this is just a first step and a concept which we wrote an AI for Earth grant, and I'm very grateful to be able to connect to this whole world of technology and Microsoft resources that are available for us now. to be able to do genomics even next level. I have a really nice video here which describes Quokka, but I think I'll have to pass it and go further on this thing that how, and I want to showcase a few other projects which has happened in the lab since the launch of DNAZOO Australia last year in July, that how we've been able to study these genomes in deep time using the power of comparative genomics and open source science. It's really, really important that we have this data available openly so that people can use it to query many different things they have in mind and they have always been chasing for. So this is Aisling Charles. She's amazing, and she worked with us. She's an undergraduate student at University of Western Australia, and she came to us, and that she's really motivated by evolution, and she got selected for the POSI supercomputing internship, which happens every summer. And we've started looking into unlocking life origins using all these genomes which we have put together at a base pair, single base pair resolution, because we can now, and we have all these genomes. So we looked into cactus, which is pretty much the only program that is available right now to be able to do non-reference based assemblies, so that there's no reference bias. We're not squeezing everything to the human genome. This has been a fascinating project, but had a bit of a thorny start in terms of taming this package onto the high performance computing environments which we work on. But stay tuned. This is progressing really, really well. So if you're doing something similar, please get in touch there. We might be able to move faster progress with other people who are putting efforts in this space as well. We've had an amazing another intern student, Tarun, from Monash University, working with Sonika Tyagi. She's an amazing collaborator, and where we tried to characterize and analyze the immune genes in all the marsupials, at how the immune genes that have been well characterized in the human, there's about 800 such genes, and how they sit in a Tamar wallaby genome, or how sort of they are arranged in other marsupial species. As we know that they're very, very unique species. So that was an interesting project, that we can look genomes in deep time and start to answer some interesting biological questions. Not just that, actually, and while we were busy all this, COVID happened, and that just, I feel it's like probably we are in a better situation right now in WA, but most of you who are joining us from eastern states are still going through a lot of damage and distress because of this COVID. But at DNAZOO, luckily we were prepared. We had a lot of bad samples in our collections. So this happened in January. I was actually in Houston lab at that point of time when this bat species were being assembled. This was the Madagascar roseate, which was actually thought to be one of the bat species from where probably SARS-CoV-2 has come into the human species. And that genome was released straight away. But not just that, all the other bat genomes which we had in our freezers, in our vault, we were able to put the chromosome length genome assembly supporting that work, including the most trafficked species on the planet, with pangolin, which happens to be also one of the species suspected that this might have come from. So this was the power when there was, all the samples were available, the collaboration network was there, the technology was there. It prepared us to be able to do and support us, to support a situation like this pandemic. Not just that, we were also able, one of the PhD student who led this study from AIDA Lab, Brian, he was working on the zoonosis, and we were able to put together a very rapid, low cost, highly sensitive whole genome sequencing method together. And we have established this method. This will be coming out as a diagnostic. It's just awaiting FDA approvals right now. So I'm not going to go into the details, but please feel free. This was released open source through bioRxiv, and the whole process, if anybody needs to use that or need that, please get in touch. It's all established on POSI Supercomputing Center. You can assemble the entire 34 MB genome in less than 1 minute. So it's really well nutted out diagnostic, which cost less than $10, and it's world's second on the list in terms of its sensitivity as approved by FDA already. So, as somebody said, humanity has squeezed the world's wildlife in a crushing grip, and what has come out of it? Lots of viruses, pandemics in the future. So I think establishing that is one of the key things that I have been aspiring for, and we welcome anybody who wants to learn these technologies at UWA to come and work with us, collaborate with us, so that we can prepare the skill sets for the future and also are in a better space to deal with these pandemics. There are global labs now. So Houston is the headquarter lab, and then we've got DNAZOO Australia at UWA. POSI is our data headquarter. We do all the storage analysis at POSI. And also we've got DNAZOO China, and there's 3 more satellite labs coming this year soon. The pandemic has delayed them, but yeah, we're hoping to have a lab in every continent soon. I was going to take you guys through the role of POSI, but I would just say that all our data storage, management, and analysis for the data which comes across the globe is happening on POSI. So it's a great thing for all of us in Australia that we have this opportunity to work with our supercomputing clusters to be able to make this impact in a global space. Everything we try to do the containerized so that it could be run in other compute environments if you've got access to in a smooth and efficient way. Everything's open source. DNAZOO methods, which I talked about, they are very beautifully described in Genome Assembly Cookbook written by Olga Dachenko. So please go and download and have a play with the code. We share all the data without restriction through the website. There's these pretty chromosomes there, which has been developed by ADAM Photos in Chicago, Washington University. Amazing resource if you're running outreach activities and trying to explain genomics to school kids. We publish genomes every week. Please subscribe the DNAZOO on Twitter if you have not. So that's where you can get all the information, or you can subscribe on the website and you can get an email about every new genome release that happens weekly. So far, these are all the amazing collaborators. Very, very, very grateful to this entire network to support the project. And also all the data has been made freely available and being pushed on NCBI as well. It's huge amounts of high-C experiments across 156 species. So please feel free to go and check that out. We're hoping that these efforts can not only aid the non-human populations, but it will actually help us understand the tree of life a little better and also evaluate it to go better in the future. As David Attenborough said, and I'm surely not comfortable when my son tells me that Tasmanian tiger is extinct and we can never see it in the zoo anymore. So are we prepared to lose more species like that? I think all that advancements which we have done is useless if we let our biodiversity fail. So with that, I would like to say greetings from DNAZOO and all the amazing corporate partners and the networks which have supported this and made this happen, and the support of University of Western Australia and all the students and team that work in our lab to make this happen. Thank you very much for listening. I'm really sorry I haven't left much time for the questions, but before I go off, I would like to say we're very, very, very grateful to everybody and many more who I couldn't fit on this slide, who've believed in us as we launched DNAZOO Australia last year in July, provided us samples and worked with us and supported us. And it's been an amazing journey. I have learned a lot from you all, and we look forward to have more people join this effort. Thank you so much. And I'm open to questions. I'll look into the Q&A tab for the questions now.
Well, thank you very much, Parwinder, for the wonderful overview of Hi-C and DNAZOO and your love of quokkas, and for sharing your mission for genomic empowerment. The urgency and the importance of the conservation of our threatened and endangered species is only growing, so we wish you all the success. We have used up our time together today. Parwinder has left her contact details on the screen here. She's very open and responsive by email and by Twitter, so please reach out to her if you have any questions that you'd like to follow up on. So that is all we have time for today. I'd like to once again express our thanks to you, Parwinder, for sharing your inspiring work, and I'll draw the presentation to a close here.
Thank you, Christina. Thank you so much for having me.
We hope you found today's presentation useful and interesting, and we also hope that you'll join us again. Finally, we'd quickly like to acknowledge our funders. The Australian Biocommons is enabled by NCRIS via Bioplatforms Australia funding. Until next time, goodbye.